Okay, thank you for the invitation to present at LabRoots. My name is Adam Burrack, and I did the work that you're about to see with my hands in the lab of Dr. Ingen Stromness at the University of Minnesota. So our specific interest is interrogating mechanisms of tumor-specific T-cell exhaustion and activity in pancreatic ductal adenocarcinoma. So to begin with, our research problem that we're addressing with our model system and then interrogating T cells within that model system is really a lack for the field of an antigen-specific T cell model to study why immunotherapy fails in pancreas cancer. The reason underlying that lack of a model is that there are vanishingly few pancreas cancer-specific T cell responses that have been characterized in patients. So we really don't understand how T cells fail to attack pancreas cancer. So in the basic immunology field, which is where I work, mouse models, there is an ovalbumin positive pancreas cancer cell line that has been made. However, these tumor cells, when they're injected into mice, are overtly rejected or they lose antigen over time. So we really can't study in this model what immunologists would call T cell exhaustion and how T cells respond to treatments like immune checkpoint blockade, sort of exemplified by PD-L1 blockade. So a potential rate limiting factor for T cell access to tumors, even if they were tumor specific, is the so-called fibroinflammatory microenvironment that characterizes pancreatic cancer, which may restrict blood flow. And because blood flow is restricted, and that's how T cells would get into the tumor beds, that may be why there are few T cells within pancreas cancer, regardless of antigen specificity. So to begin to address these problems in a basic model in mice, we transduced several different KPC mouse-derived cell lines, which is what these KPC 1, 2, 3 are on the left, with a construct containing click beetle red luciferase as well as GFP. So click beetle red facilitates tumor imaging by luminescence over time as the tumor grows. And when we made these cell lines, when we transduced these cell lines with this construct, GFP or FITC expression is how we sorted our cells that were transduced. So the cell line that we used primarily in this Cell Reports paper that was published in 2019 is this KPC2, and then this A line here, the top red on that graph on the left. That cell line was well transduced with our construct, so we could orthotopically inject this cell line into the pancreas of mice and visualize tumor cells over time. And the main point here of showing the immunofluorescence imaging on the right is in this orthotopic model on the bottom, we see that there is a degree of fibroinflammation that does develop over time, over several weeks following tumor cell injection. Alpha smooth muscle actin is one of the markers of the fibroblasts in these orthotopic tumors. We also have F480 positive macrophages in red, some CD8 T cells in yellow, and some neutrophils by Ly6G staining in purple in these tumors. So a range of immune infiltrate. The tumors grew well, and we have a model system. One defining factor of pancreas cancer clinically is that it resists immune checkpoint blockade. PD-1 and PD-L1 were tried in initial studies in 2012 and 2013, and patients resisted these therapies. However, in our model system where the tumor cells express this click beetle GFP construct, tumor luminescence decreases following these treatments, shown on the left. We have very bright tumors at day 7, and then following treatment, which is initiated at day 7, and then it's another treatment on day 10 and day 12 after tumor cell injection. These tumors have less luminescence following that treatment. Whether we block PD-L1 on the bottom middle or PD-1 on the bottom right, luminescence goes down over time, and at 3 weeks after tumor cell injection, we have significantly less tumor luminescence, consistent with a tumor-specific T cell response, we would hope. So another observation that is consistent with a T cell response against our model antigen potentially, or at least against our tumor specifically, is the presence of CD8 T cells by immunofluorescence staining in these tumors, as noted by the yellow staining in these images and the arrows pointing to those yellow cells quantified on the right. Even without the PD-L1 treatment, in the number of CD8 cells per field of view graph on the middle right, we have a large number of CD8 cells per field of view without PD-L1 treatment, and then in the green, we still have a large number of CD8 cells per field of view. In addition, below that, on the bottom right, we have relatively high intensity for PD-L1 staining in the tumors in untreated mice, just with the KPC2A cell line shown in red, and that intensity of PD-L1 staining is maintained following PD-L1 blockade. So this would suggest that there's probably a fair degree of interferon gamma expression in these tumors by CD8 T cells, which is promoting that PD-L1 expression by either tumor cells or tumor-associated myeloid cells. And again, that would be consistent with a tumor-specific CD8 T cell response occurring. Strikingly, and the reason this paper was well received by reviewers, was PD-L1 blockade, presumably to this model antigen, prolonged mouse survival, as shown in the survival curve on the bottom left of this slide. We can also quantify tumor volume by ultrasound imaging, in addition to luminescence, which is the only method that would quantify tumor size in the absence of this luminescent molecule. So in cell lines that either don't express the construct at all, this parental KPC2 line in the top middle, or the GFP only control cell line, our tumor volumes did not decrease following PD-L1 treatment, PD-L1 blockade. Whereas the click beetle expressing line, those tumors were smaller following PD-L1 blockade. The tumor weights at day 22 were consistent with this smaller volume in PD-L1 treated mice that received our KPC2A cell line. Oops, sorry. In contrast, PD-L1 blockade treatment, as I mentioned earlier, did not prolong mouse survival, did not decrease tumor volume or tumor weight in the absence of this click beetle molecule. So that would suggest that we have a specific T cell response to some peptide derived from that click beetle protein. So our next step was to figure out, using algorithms to predict peptides and then an empirical test of the highest scoring peptides derived from that protein, what our immunodominant class 1 MHC class 1 restricted peptide was that CD8 T cells were responding to. So these, in the top left, the numbers on the bottom of this graph are the initial amino acid of the peptides that we tested. So this peptide beginning at amino acid 101 of the protein was our winner in this competitive assay in vivo. We saw the most interferon gamma production to that peptide. So we went ahead and made an MHC class 1 restricted H2D of B is the class 1 that we were testing here. Restricted tetramer reagent and could detect specific cells in vaccinated mice and also in tumor-bearing mice. So following this discovery, we had a handle to study tumor-specific cells in pancreas cancer. So we did the first thing a lot of immunologists think of is immune responses sort of evolve and happen over time. So we did a time course. We looked at these tetramer binding cells in the blood, the spleen, and the tumor on day 7, day 14, day 22, and in mice that were treated with PD-L1 blockade, day 43 after tumor cell injection. And when we gated on our CD8 positive T cells, we found robust populations of tetramer binding cells at all of these time points and in all of these locations. Consistent with the premise that T cells get into these tumors just fine. The tumor itself does not restrict T cell infiltration. And even in untreated mice with large tumors, at day 22, the control, there were between 30 and 40% of the CD8 T cells found our tetramer. So there are in this model, plenty of tumor-specific T cells in the tumor, and the tumor continues to grow. So that would suggest that the cells in the untreated mice get exhausted, or something similar where they cannot kill the tumor cells. And with PD-L1 blockade, that process is perturbed somehow. So one possibility would be canonical T cell exhaustion, which could be defined as co-expression of the inhibitory markers PD-1, LAG-3, TIM-3, and TIGIT on our tetramer binding cells. And these pie charts are probably the most useful way to look at this. We think in the tumor, in either untreated mice, the darker these pie chart areas, all four of these markers are expressed. Slightly lighter 3, lighter yet 2, just a little bit gray is 1, and then the white is none of these markers expressed. And the entire pie chart represents all of our tetramer binding T cells. So in the tumor, in untreated mice at late time points, we have about half of our cells that bind the tetramer express either 3 or 4 of these canonical exhaustion markers. Whereas if you compare on the right, PD-L1 treated mice outlined with the red outline of the circle, the majority of the tetramer binding cells at the same time point are expressing either 0 or 1 of those canonical exhaustion markers. And in this case, the 1 tended to be PD-1, which is generally thought of as more specific to T cell antigen encounter rather than exhaustion per se. In our hands in this model, the combination of PD-1 with LAG-3 expression on the same T cell appears to correlate better with an exhausted phenotype than just PD-1 itself. In addition, in the anti-PD-L1 treated mice, the peptide-specific cells retain the ability to produce interferon gamma in an ex vivo restimulation experiment more effectively than in untreated mice. This is quantified on the right. So we're comparing untreated mice to PD-L1 blockade treated mice. This is at the 3 weeks after tumor cell injection time point. And look in particular here at the cells isolated from these tumors. We have a higher proportion of CD8 T cells able to produce interferon gamma in response to click beetle peptide restimulation in a cell culture dish for 4 hours. So that would suggest that treatment helps T cells either one, retain function, or B, potentially a new wave of effector T cells is stimulated from the spleens of these mice. Okay, so the next observation was, I mentioned earlier that LAG-3 may be a better marker of T cell exhaustion than PD-1. Our best marker of T cell activation, turns out in this system, is KLRG1 expression. And it appears to be more or less a binary choice for an individual T cell to become either a LAG-3 positive exhausted cell or a KLRG1 positive effector cell following treatment. And at about the time that I was going to start combining therapies, a paper came out in a different model of cancer. It was a melanoma paper where they combined PD-1 blockade with PD-L1 blockade with a hypothesis that PD-1 blockade would activate T cells and PD-L1 blockade would activate the macrophages. And in that paper, they saw benefit of this dual therapy. And I thought that would be cool to try in this system. So we did. And I was agnostic on whether PD-L1 blockade was activating macrophages or not, but we gave the dual therapy a shot. And in the green line here, you can see that we cured half of those mice. That was really satisfying. We did a lot of treatments here. We treated these mice with both PD-1 and PD-L1 blockade from day 7 to day 28 after tumor injection. So that was a lot of antibody and a lot of treatments, but we did cure half the mice, and we promoted the KLRG1 expression over LAG-3 expression, quantified in this bottom row of plots. So that was really satisfying to cure mice of a model antigen expressing pancreas cancer model. So one question we could ask, having developed this model where we can cure pancreas cancer, is is starting therapy at day 7 after tumor cell injection required, or could we delay therapy? Because as the audience is probably aware, pancreas cancer is generally diagnosed pretty late in the process, stage 3, 4. So in this model, it turns out that delaying therapy is a big problem. On the right, if we delay our dual therapy, which can cure half the mice, if we delay the start of that by 1 week and do that first treatment on day 14 instead of day 7, it looks much more like the survival curve of our single therapy started earlier. And if we delay the single therapy start date by a week, it is essentially no benefit to the mice. So timing matters. So to summarize this first part, our model antigen represents a very high affinity peptide interaction with MHC, which primes a really strong T cell response. So some further questions that we can ask is what specifically is driving intratumor T cell exhaustion? What protein expression identifies this state on the T cells? And can we improve, with this knowledge, can we improve our success rate of our therapy? So this moves into another publication that just came out recently in the Journal of Immunology, where we interrogate these questions. And we did, not surprisingly for immunologists, another time course. So in this case, we're looking at expression of different proteins on our tetramer-binding T cells in the spleen and the tumor over time. So on the left is a visual summary of this. Spleen cells that bind tetramer are blue, tumor in red. And we're looking at tox expression, which signifies antigen experience or perhaps ongoing antigen experience, and granzyme B, which is one of your key contact-mediated factors that T cells express that cause target cell death. So at day 7, we have a lot of both of these in the tumor. At day 14, we've maintained tox but are losing granzyme B, and at day 21, we have maintained tox but we have lost granzyme B. So that's essentially the story. Our T cells in the tumor continue to express antigen indicated by tox, but they lose granzyme B expression, so they can no longer kill tumor cells. In addition, on the right, on the bottom graph, in the tumor, the proportion of tetramer-binding cells that are able to express interferon gamma also goes down. And interferon gamma is another critical cytolytic molecule that CD8 T cells express. So 2 different ways to kill tumors, and our T cells lose both of them over time. So in addition to tox maintenance and granzyme B loss and interferon gamma loss over time, another component of T cell exhaustion in the tumors in our model system is T cells acquire GFP expression, or not GFP expression, IL-10 expression. So our tetramer-binding T cells begin to express IL-10 over time, as shown on the right, using IL-10 reporter mice. So it's a GFP reporter mouse. The cells that are GFP positive, those in the tumor, tend to express PD-1, LAG-3, TIGIT, TIM-3, our canonical exhaustion signature from the first paper. So as these cells become exhausted, they are maintaining tox, losing granzyme, losing interferon gamma, gaining IL-10. So what's driving this? The first thing we tried was to block all these co-inhibitory markers in combination with PD-L1 blockade or individually. And clearly from the survival curve, that did not help. So messing with the phenotype at the end when they express all of these exhaustion markers is not effective. So we need to move upstream in the process. And one way that we could go about trying to interrogate what that was driving all of these exhaustion markers is to look in some of the clinical databases for either cytokines or chemokines or growth factors that either the tumor cells themselves express or the myeloid cells in the tumors express that correlate with impaired patient survival. So we're trying to find something that's clinically relevant here. And it's really hard to make pancreas cancer survival worse. I spent a lot of time in the database trying to find something. IL-27 is one of those things. So high expression of IL-27 P28 within pancreas cancer actually makes pancreas cancer survival worse. So lucky for us, there's a reporter mouse for IL-27. Cells that are expressing IL-27 gene, those cells also express GFP. And as a proportion of CD45 positive immune cells, the proportion making IL-27 goes up over time in the tumors. suggesting that our mouse model may be reflecting what's happening in the human disease where IL-27 is not useful. And this is weird and interesting because IL-27 is critical for T-cell priming and is actually really important in the response elicited by type 1 dendritic cells after vaccination, which is a topic for another time, but is why I was aware of this field. IL-27 is really important for T-cell priming and memory formation, but too much may be a bad thing. We did another time course, and we characterized which myeloid cells in this case, or non-T cells that are in the tumor, are expressing IL-27 at either day 7, 14, or 21 after tumor cell injection. And at day 7, it was clearly granulocytes or neutrophils that are the main source of IL-27. At day 14, it was a mix of type 2 dendritic cells, which in this case, in our gating scheme, are SIRP alpha positive, along with macrophages, tumor-associated myeloid macrophages, and some other cells, including type 1 dendritic cells at day 14, and then several cell types at day 21. So essentially, our T cells that go into our tumors, primed and ready to go from the spleen, encounter just an ocean of IL-27 and probably a bunch of other suppressive cytokines, and that may be part of T cell exhaustion developing over time. CD40 agonist is in the clinic in clinical trials to treat pancreas cancer and some other malignancies. So we thought it was rational to try one treatment with CD40 agonist at day 7 after tumor cell injection and see what that did to IL-27 expression by various immune cell types. And long story short, we saw a big decrease in IL-27 signal with CD40 agonist, single therapy, but not with our PD-L1 single therapy, which we already knew prolonged survival and had effects on T cells. So it appears here from these data that CD40 agonist is affecting the myeloid cells qualitatively. In addition, IL-10 expression is downstream of IL-27 receptor signaling. So it was rational, in our minds at least, to think that IL-27 exposure of T cells could have been driving that IL-10 expression that we saw earlier with the reporter mouse. And it turned out that CD40 agonist treatment on the left decreased the proportion of CD8 T cells that were IL-10 positive in the IL-10 reporter mouse, whereas PD-L1 blockade did not. In addition, CD40 agonist on the right, shown on the right, promoted granzyme B expression by our tetramer binding T cells, which again, PD-L1 blockade did not. So two different therapies, both effective to a degree, doing very different things. So we did the immunologist thing and we put them together. Very simple treatment schematic here. Our 3 treatments of PD-L1 blockade at day 7, 10, and 12, 1 treatment of CD40 agonist, and we cured 10 out of 16 mice with the dual therapy group, which is really, really satisfying. IVUS imaging shown on the right. We essentially don't see these tumors luminescent at all after we initiate therapy with this dual therapy. It's gone until it comes back much later in the mice where it does come back. And in many of the mice, it does not. Through some phenotypic analysis, it appears that CD40 agonist promotes what we're calling in this publication a short-lived effector T cell phenotype. So in the blue dots for CD40 agonist single therapy at day 14, we're seeing a lot of those KLRG1 positive cells. We're seeing a decrease in TOX expression. We're seeing a decrease in PD-1 expression, and we're seeing a decrease in LAG-3 expression on our tetramer binding cells. So again, remember the combination of PD-1 and LAG-3 is our exhaustion phenotype, and we're seeing essentially fewer of those and more of these KLRG1 positive cells earlier, consistent with a really strong response, really quick, and you just get rid of the tumor. At later time points, like 3 months after tumor cell injection, when the mice have had no luminescence for 2 months or so, we see tetramer binding cells not only in the pancreas and the spleen, which you would expect, but also in sites of potential tumor metastasis, the liver and the lung. Shown on the left, we had over 10% of the CD8 T cells by flow cytometry in those locations found the tetramer. But interestingly, In the pancreas is where our tetramer binding cells express the residency markers CD49A and CD103. In addition, if we re-challenge these mice that had cured a previous tumor with a second tumor in the pancreas 100 days after the first tumor, the tetramer binding cells expanded 10-fold to 100-fold in the blood, the spleen, and the pancreas, consistent with really robust T-cell memory responses. Two straightforward things that classically trained immunologists would ask in this setting is what effector molecules is this response dependent on? And the first two that came to mind for us were interferon gamma and TNF alpha, because those are the cytokines that we look for in our peptide restimulation experiments. And for example, transplant rejection requires essentially both of these. Interferon gamma in the blue dotted lines are untreated mice. The blue solid line is our dual treated mice. You'll see the blue solid line goes straight down at day 40, which means that our dual therapy had very little benefit in mice that did not have interferon gamma receptor signaling, suggesting that this is an absolute requirement for our effector T-cell response, as probably expected. In sharp contrast, the green solid line is TNF receptor 1 knockout mice. And in those mice, we actually had 4 out of 4 mice cured and remained cured with our dual therapy. So this would suggest that somehow TNF receptor 1 signaling may be impairing either the effector response or the memory response to these tumors. So the last experiment that I'll present, this sequence is looking at the memory T cells in either our regular B6 mice on the top 2 panels or TNF receptor 1 knockout mice on the bottom 2 panels. And what we see immediately are a higher proportion of CD62 ligand CD44 double positive so-called central memory cells in the knockout mice on the bottom left, and co-expression on the right of CXCR3 and KLRG1 on some of those cells in the receptor knockout mice. So this is something we're pursuing now, and it really suggests that there may be qualitative differences in memory T cell generation when you don't have TNF receptor signaling. That's one of the main thrusts of an R01 that we just started. So that'll be really exciting to interrogate moving forward. So to summarize quick, we show in this second paper that tumor-specific T cells lose granzyme B and interferon gamma within the tumor specifically, while they maintain tox expression, suggesting that they're continuing to see antigen, and they gain IL-10. We found that CD40 agonist ablates chronic myeloid cell IL-27 expression, along with T cell IL-10 expression, and promotes T cell granzyme B and KLRG1 expression, consistent with a short-lived effector phenotype. And given these really broad differences in the effects of CD40 agonist and PD-L1 blockade, it is not surprising that these two therapies synergize. And further benefit in the absence of TNF receptor signaling, which is really intriguing and, again, a main direction for us moving forward. A couple quick acknowledgments. Firstly, and most importantly, Ingen Stromness for the opportunity to do the work in her lab, and everybody in the lab, too many to thank, and our new R01 on this project. Thank you again for the opportunity. I'll be happy to answer any questions.